All right, welcome back, biology students. We are going into our next set of notes. So today we're going to be looking at something called Punnett squares. Punnett squares are a way for us to find out the probability of what types of crosses we're going to get. So today's learning target is I can predict offspring, phenotypes, and genotypes by using probability. So your bell work question today is, what does probability mean? So answer that question now in the ed puzzle. All right, so Punnett squares. Punnett squares is a grid system for predicting all possible genotypes that are resulting from a cross. The axes represent the possible gametes of each parent, and the boxes in the grid show the possible genotypes of the offspring. The Punnett square yields the ratio of possible genotypes and phenotypes, so their actual genes and what they'll actually look like. Here's an example of a Punnett square. You can see here we have parent one up here, and these are the alleles from parent one. So this parent is heterozygous. They have one big letter, one small letter. Parent two is over here. They also are heterozygous with one big letter and one little letter. These are the alleles for the trait. Each parent has one dominant allele and one recessive allele. And then these boxes represent the possible outcomes we could get from these two parents, because we don't know which of these letters are going to go into the gamete. Either one could go in. There are two types of Punnett squares that we're going to be dealing with. The first is called a monohybrid cross, which means we're only examining the inheritance of only one trait at a time. Like in this first example here, you can see we're only using the letter Y, uppercase and lowercase, and that's to represent the color of a pea plant, of the peas in the pea plant. So dominant is yellow and recessive is green. A dihybrid cross, on the other hand, involves two traits at a time. You really have to get good at a monohybrid cross first before you can do a dihybrid. But here's an example of a dihybrid cross where we see that we actually have two alleles per box. So our grid actually expands by 4 times. So this has 4 boxes and this has 16. So it's 4 times the amount of a monohybrid cross because we're adding another whole nother trait. And so you can see here, the capital Y's still mean yellow, the lowercase y's still mean green, but then we also have this variable r, which is representing whether or not the peas are round or if they're bumpy. Okay, and those are the traits for that one. When showing the ratio of Punnett squares, you must follow this order. For genotype, homozygous dominant, heterozygous, and then homozygous recessive. That's where we would put the numbers. So if it's like a 1-to-1-to-1 ratio, or a 2-to-1-to-1, or a 1-to-2-to-1, that's how we put them. We put them in order of homozygous dominant first, heterozygous second, homozygous recessive third. So for example, this one, we have a phenotype that's, if we're looking at phenotype, then we just do dominant recessive. So for example, in this one here, this box would be big Y, big Y. So that's homozygous dominant. This box and this box, though, are going to be big Y, little y. So those are both heterozygous. And then this one here is homozygous recessive with 2 little y's. So there's one homozygous dominant, so 1. There's 2 heterozygous, 2, and 1 homozygous recessive. So the ratio in this particular example would be a 1-to-2-to-1 ratio. Now, as far as phenotype, 3 of them are yellow, 1 is green. So this is a 3 dominant to 1 green recessive ratio, 3-to-1 ratio. So let's take a look at some simple dominance. So simple dominance occurs when an inherited trait is coded for by a single gene, and that gene has 2 versions or alleles, the dominant version and the recessive version. Like this example here, yellow is dominant, green is recessive. So we can go through different generations if we know our starting point. So for example, in this example with the pea plant flower color, we have a homozygous dominant as one parent. That's this one here, big F, to show purple flowers. And then a homozygous recessive is the other parent, little f, to represent white flowers. So the genotypes of the parents, these are the first generation, would be the first genotype would be big F, that would go here. And the other parent is little f, that would go here. And then the F1 genotype ratio, well, here are all of the possible offspring. Notice every single one of these offspring, based on these two parents, is heterozygous, big F, f. So the genotype ratio here would be 0 to 4 to 0. All of them, 100% of them are going to be heterozygous. And then the phenotype ratio would be, again, 4 to 0. It's 100% that they're all going to be purple. Okay, let's try a different one. Now, if we were to take a heterozygous and mix it with a heterozygous, so now both of the parents are heterozygous rather than being a homozygous dominant, homozygous recessive. So the genotypes of the parents are big F, little f for parent number 1, big F, little f for parent number 2. But then the genotype ratios, there's one homozygous dominant, so 1. There are 2 heterozygous, so 2. And then 1 homozygous recessive, 1. So the ratio is 1 to 2 to 1. The phenotype ratio, however, is 3 dominant purples, so 3 to 1 white, 3 to 1 ratio. So if these were to mate, we would expect that we would get about 3 purple plants for every 1 plant, for every 1 white plant that's born. Let's try a heterozygous versus a homozygous recessive. So this first parent genotype, little f, we're crossing it with a heterozygous parent, big F, and then the genotype ratios would be 0 are homozygous dominant, 2 of them are heterozygous, big F, and then 2 of them are homozygous recessive. So it's a 0 to 2 to 2 ratio. The phenotype ratios, on the other hand, would be 2 purples to 2 whites, so 2 to 2, or in this case, 1 to 1 ratio if we take it to its lowest common denominator. When we do Punnett squares with simple dominants, it's always good to do a test cross, and this is to determine the unknown genetic makeup of the dominant phenotype organism, since it can be homozygous dominant or heterozygous. So for example, if we know that, let's say, black fur or hair on a sheep is dominant, but we don't know if this sheep is big B, or if it's heterozygous, big B, little b, well, then we cross it with one that we know is recessive, little b. And so if we cross it and we get certain combinations, we're going to know what we're dealing with. So for example, if we do the cross and we get 100% black sheep, then we know that this parent was big B, because we crossed it with a parent that we knew, and the only way we're going to get 100% black is if this one is homozygous dominant. But then if we do this cross, we see in this example, only half of them would be black and half of them would be white. And so that tells us that this parent then would be heterozygous, big B, little b. Also, with Punnett squares and simple dominants and dihybrid crosses, Mendel's dihybrid crosses for biology 1 are just with heterozygous organisms, and they're always going to yield a 9 to 3 to 3 to 1 phenotypic ratio as we go throughout this, and we'll practice this more in class. This will be a little harder to understand until you see some practical examples in action. Okay, so here's your EOC question for the day, and I want you to answer that here at the end of this UD puzzle. Thanks for watching, and good luck.